Hello, how's everyone doing? I'm here to start the next section in zoology. So, first section was just kind of cell biology, molecular biology. Second section was genetics and evolution and natural selection. Now we're going to shift more into sort of specific things that are maybe unique to animals and kind of dial into some details about some of the the processes and things that go on in animals and get them in a little bit more detail. And so we're going to start with the reproductive process, and we're going to talk about several different examples here. So why is this important? We've already talked about meiosis and genetics, and that's tightly tied to reproduction, obviously. Passing traits from parents to offspring, that's the cornerstone of evolution and natural selection, right? Passing traits to your offspring is fundamental to all organisms, and continuing your lineage is just an innate something innate to living things, right? And so understanding this process is important to understanding biology in animals. So we're going to talk about the nature of the reproductive process, some different ways that sex is determined, how gametes are formed, and then very briefly reproductive patterns. So again, one of the things that I love about biology is that there's just so much variety, and there's so much, I don't want to say creativity, but there's just so many different ways to accomplish the same goal. And it's interesting, these different solutions to common problems. And so we see a huge variety in the ways that organisms and animals reproduce, right? There's no such thing as a typical reproductive method. So, we tend to judge things by our own species, and we tend to compare everything to how we do it as human beings, but there's just no typical way. I always say, if you can imagine it, some organism somewhere does it, and it's certainly true with the reproductive process. And so, we've touched on some of these before. The very basic difference in different strategies is asexual versus sexual reproduction, right? So asexual is a clone of the parent, and it does not require the production of gametes. And so there are many ways of doing this. We've touched on some, but here's a couple examples from your book. Binary fission, you got the paramecium here. Again, not an animal. But it just simply reproduces by splitting in two. That's what bacteria do. Budding by this hydra, which is an animal. Just a small version of the parent starts to grow and then eventually breaks loose. There are other forms of asexual reproduction in the animal world. So sexual reproduction is not a clone of the parents. Now remember, the offspring in sexually reproducing organisms, they have the same traits as their parents, right? That's natural selection chooses, or those organisms that are well suited to an environment tend to stick around and reproduce. And so their traits tend to stick around, but sexual reproduction gives you new combinations of those traits. Of course, this is accomplished through the formation of gametes and meiosis. The sex cells are also called germ cells, or the germ line. This is the opposite of somatic cells, or somatic or body cells. So the germ cells are the cells that make gametes. Now they can do both mitosis and meiosis. The germ cells themselves need to replicate, and that requires mitosis, but then they have the ability to go through meiosis to create the gametes. The gametes are going to form, are going to combine to form a zygote. And so there's all kinds of sexual reproduction. And again, it's just fascinating when you study the animal world, all the different ways to reproduce. Bisexual or biparental, so that's what we do. That's males and females. But hermaphroditism, parthenogenesis, these are other ways that you can sexually reproduce. So we're most familiar with biparental, which we can also call dioecious. Di is Greek for two. So you've got two sexes, and each produces only one kind of gamete. And so the females produce eggs, which are also called ova. And eggs are characterized by being large and non-motile, and you don't produce many of them, So, eggs don't really move. They're non-motile. Males produce sperm or spermatozoa. And these are small, and you produce millions of them, and they're motile. They can move. And so that's general differences between the gametes. And so, again, you're familiar with this, right? A male and a female each go through meiosis to produce their respective gametes. And then after mating, the gametes fuse, and that's a zygote. And then we've talked about how the zygote develops into a blastula in animals, and then, in some animals it gastrulates and so on. And then you have mitosis, which produces the growing organism, which eventually, when it reaches sexual maturity, can then also go through meiosis and so on. Now, in hermaphroditism, instead of dioecious, this is known as monoecious, right? One, mono being one. And so a hermaphrodite has both the male and the female reproductive organs. Now, sometimes the organism can self-fertilize. So if you've got both the male and the female organs, you can fertilize your own eggs with your own sperm. And sometimes they fertilize others. And so you can have both simultaneous hermaphrodites and sequential hermaphrodites. So simultaneous hermaphrodites means you've got both male and female gonads at the same time. And so again, you could fertilize yourself, or you could exchange sperm and egg with other simultaneous hermaphrodites. Sequential hermaphrodites start out as one sex and then can turn into the other sex. And in some species can turn back. And we'll talk about that in a second. And so later on in lab, we're going to talk about earthworms, and they are simultaneous hermaphrodites. They have both male and female reproductive organs. They produce, every earthworm produces both sperm and egg, and you can see here how they're mating. And there's something called the sperm groove, and they release sperm, and it travels down that groove. And so every worm can fertilize every other worm and be fertilized by every other worm. This is a species of fish that is a sequential hermaphrodite. It starts out as female and then eventually can turn into a male. We'll talk about this in more detail. This is very common in fish species, especially marine fish species. Okay, now the last type of sexual reproduction that I mentioned is parthenogenesis. And this is when an organism can develop from an unfertilized egg. So you're still producing eggs and you're still producing sperm, but the egg can develop without sperm. No male is necessary. Now there are also interesting examples of this where males are necessary and they'll mount the female and they'll go through the mating process, and that stimulates the female to produce and release an egg and the egg to start developing, but you don't actually need the sperm. There's all, again, if you can imagine it, some organism somewhere does it. Now, this egg can be diploid and just start reproducing or start dividing and produce a new organism. And if that's the case, then it would be a clone of the mother. And this would not count as sexual reproduction. That would actually be a form of asexual reproduction. But it could be a typical egg that's formed from meiosis, and so it's haploid. And if that egg is going to develop into an organism, it needs to double its DNA and then start dividing. And so it starts as haploid, doubles the DNA, now it's diploid, now you can develop into another organism parthenogenically. And so, we talked about some ways that you can double that DNA, right? Just no cytokinesis, you can replicate the DNA and then have no cytokinesis, for example, or something like that, to double the DNA. In this case, it wouldn't be a clone, because when you go through meiosis, those homologous chromosomes, the paternal and maternal chromosome line up randomly and get separated randomly. And so then whatever gamete forms, and then you double that DNA, it would actually not be a clone of the mother. There's also something called haplodiploidy, where a fertilized egg will become female and an unfertilized egg will become male. You're going to see this in a lot of insects and things. So again, it's just fascinating. There's just so many, there's no typical way to reproduce. Now, this is a great example. This is from the Louisville Zoo. So this is just up the road. This is a python. I think she's a python named Thelma that spontaneously produced 6 babies. Now I think she's 20 years old. She'd been in the Louisville Zoo for decades, all by herself. There's no other males. She's not with any other snakes. And so when she laid eggs and raised babies, actually I don't know enough about reptiles, I'm sorry. And I don't know if they lay eggs or if they have live birth, but at any rate, she produced 6 babies that lived. And so of course they were stunned. They're like, how did this, what? And DNA testing showed that they were, again, I'm not sure if they were clones of her, but they were all just her DNA. She's never been in contact with a male, and she's not been near any male. So here you have, this is the first example, this is the first published example of vertebrate parthenogenesis. And so, again, think about it. I mean, this is your classic virgin birth, right? This is a vertebrate that produced babies and did not require a male. And so that seems like something that people would say would be impossible, but this is just up the road again. This is in Louisville and showed it's possible. So it's fascinating. Probably that this is something, if I had to guess, and I'm not the only one that guesses this, but I would speculate that this ability is retained by many organisms, because this would have a huge selective advantage. That if you were a female that was in a situation where you couldn't find any males for a long time, the ability to still produce offspring would be an advantage. So I imagine that we're going to see more reports of this as people watch for it more. Okay, so we've got both asexual and sexual reproduction. Sexual reproduction is really common in the animal world. In fact, it's more common than we would expect. And you say, well, why wouldn't you expect it? Well, there's some huge downsides to sexual reproduction. There's some huge prices to pay for sexual reproduction. And yet we see it everywhere. Well, what are some of the downsides? Well, this is not an exhaustive list. But if you think about it, for example, finding mates and competing for mates, that requires a huge amount of time and energy. Think about how many animals spend so much energy trying to attract a mate and compete for a mate, which you wouldn't have to do if you reproduced asexually. There are a lot of sexual characteristics that hurt your survival. Classic example is the peacock's tail, right? The male peacock tail is this huge ornate thing. Does that improve its survival? Does that help the male peacock fly? Does that help the male peacock hide from predators? It does quite the opposite. That huge tail lowers his ability to survive, but it's preferred by female. It helps him reproduce. And so, this is called sexual selection. But again, you're paying a huge price in survival just to be able to reproduce sexually. Males and females need to find each other. And so this is something you see in a lot of, deep ocean fish and things where the species are so spread out, and you've got to find the opposite sex, and you got to find them at the right time when you're ready to mate. Which is kind of my next point here. You must coordinate to produce zygotes. And so it's not just that you got to find the opposite sex, but you both have to be ready to mate. Another disadvantage is that it disrupts the parent's genetic makeup. So remember, we said that, the traits get passed on to the offspring, but sexual reproduction recombines those traits in different ways. Now think about it. If you're an organism that is well suited to the environment, so well suited to the environment that you have survived and you're ready to reproduce, that means that you are very well suited. Your DNA is well suited for this particular environment. And so it would seem to make sense that you would want your offspring to have that exact same DNA. If it worked for you, why not give it to your offspring? But sexual reproduction doesn't do that. Sexual reproduction retains the traits, but it rearranges them. Finally, again, energetically, males are a waste. Lots of males do not reproduce. And so if you're looking at your lineage, and if you're looking at your survival and then reproduction, and then, half of your offspring are male, and many of them will never reproduce, that's a huge investment of energy for no return. So these are all big downsides. So then why do we see it? Why do we see sexual reproduction so much? Well, we've hinted at this a lot when we talked about evolution and natural selection. It disrupts the parent's genetic makeup, but that's a good thing. It retains the traits, but it combines them in different ways. And so you have more variability in offspring. Remember, in natural selection, variability is that first thing we've got to have natural selection. And so this increases the chances of your offspring surviving a changing environment. And that is the reality in nature, is that although an organism is well suited for its environment today, environments are always changing. Not just the temperature, of course that's true. Not just the abiotic environment, but the biotic environment. All organisms live in a community with other organisms who are all also evolving. And so the rule is that organisms live in a changing environment. And so even though you're well suited for this environment, your offspring, you might not be well suited for the next environment. And so you want to pass your traits onto your offspring, but have lots of variability, so at least some of your offspring have a good chance of surviving in a changing environment. And so that seems to be a driver. That's why sex is so popular relative to asexual reproduction. A good example of how these two can combine to make even, you can kind of get the best of both worlds is in Daphnia. Daphnia is a common zooplankton, small organism that lives in aquatic systems. It's a crustacean. And they reproduce parthenogenically when the environment is stable. If the environment is stable, then it makes sense to reproduce asexually. But they reproduce sexually in a changing environment. So let's take a look at that. So here we see the Daphnia life cycle, and you can see that the Daphnia can reproduce parthenogenically and produce mostly daughters, and they're not going to have as much genetic variability. They're going to be more similar to the mother. And so they can either be clones of the mother or very similar to the mother, but they're more similar than what you would expect through sexual reproduction. But they do that when the environment's not changing. But then when the environment starts to change, when the water starts to warm up or cool down or something, then they start to produce parthenogenic sons, and then the sons and daughters get together and start to go through sexual reproduction, which is going to create these resting, the sexual egg here, they call it a resting egg. It goes into diapause. They produce these eggs sexually, so there's much more genetic variability, and these eggs are actually designed to just sit to the bottom and ride out and wait, and they can wait for a long time until the environment gets more amenable, and then the process starts again. So it's kind of getting the best of both worlds. Okay, so let's talk about how sex is determined in different animals. So in mammals, there's a gene on the Y chromosome called the SRY gene. And this gene causes germ cells to develop into testes. And so the end result of this SRY gene is to produce testes in the growing embryo. Testes produce testosterone, a hormone. And so the presence of this hormone masculinizes the embryo. Remember that hormones are chemical signals that travel all throughout the body. And so by releasing testosterone, it can bathe all the cells, and so any cell that has a receptor for testosterone is going to be influenced by that. And so it causes the embryo to develop masculine characteristics, right? Form a penis, a scrotum, stops breast development, masculinizes the brain. What's interesting is that the testosterone gets turned in to estrogen, which is the female sex hormone. And then it's actually the presence of estrogen that causes the brain to masculinize. That's crazy. But, again, to me this is fascinating, all these steps that are involved to develop the humans in the way that they develop, right? It seems like the default is to be a female, but the presence of a Y chromosome with this SRY gene overrides this. But then you could have situations where, if you had a mutation in the SRY gene, you could be XY but still be a female because that SRY gene is not functioning, right? So you can see how, there's a spectrum of different ways that you can produce different sexes. So since estrogen masculinizes the developing brain, why don't female embryos, XX females, why don't they become masculinized? Because they're already producing tons of estrogen. And it's probably because their brain cells develop in a way that they don't have receptors for estrogen. And so even though they're bathed in estrogen, they don't have the receptors. And that's the key to hormones, is that the hormone goes everywhere in the body, but it only affects those cells that have receptors for that hormone. And so just by not having those receptors in the brain, the female brain does not become masculinized by the female sex hormone. Cool stuff. And again, if you can imagine it, some animal somewhere does it. Here's the duck-billed platypus, which is a mammal. And it has 5 pairs of sex chromosomes. And during meiosis, they form a chain, and somehow the development of this chain determines the sex. And I don't really know how this works, but again, it's just, we like to think that we're typical, right? This XY system is typical, and we are not. There's all kinds of ways to get different sexes. What's also interesting about the platypus is that they have this DMRT1 gene that determines sex, but this is the same gene that determines sex in birds. And so, we got it from they got it from a common ancestor, and like the rest of the mammals, we stopped using it, but they continue to use it. But of course, the platypus is an egg-laying mammal, which is unique in and of itself. So anyway, just cool. Drosophila, you're familiar with Drosophila, fruit flies, right? They use the ratio of X chromosomes to autosomes to determine sex. So they don't have many chromosomes. And so if they have 1 X chromosome and 2 autosomes, the regular body chromosomes, the non-sex chromosomes, if they have 1 X and 2 autosomes, that Drosophila will be male. If they have 2 X and 2 autosomes, that Drosophila will become female. And so it's just about the dosage of that X chromosome. And so, it might be that there's a female producing factor on the X chromosome and a male producing factor on the autosomes. And so You know, if you only have one X chromosome, that can't override the male producing factor, whereas if you have two, it can. That's a possibility. You know, I'm not a Drosophila person. Anyway, fascinating. Now, genetics is not the only way to determine sex. Some animals use their environment, and so the environment determines the sex. A classic example of this is reptiles. Lots of reptiles use the temperature of their nest to determine what sex the babies are going to be. Some fish also use other environmental clues, other things going on in their environment, to determine what sex they're going to be. So here's a figure from your book showing several different ways that reptiles do this. And so you can see that for lizards and alligators, if the nest is very cool, then you get mostly females. And as the nest warms up, you get a higher percentage of males. And if the nest is really warm, they're all male. Whereas turtles do just the opposite. And leopards, geckos, stepping turtles, crocodiles, they do kind of a, you know, an in-between. Like at moderate temperatures, you get all male, but at very cold or very warm, you get more females. And so it's got nothing to do with chromosomes. That's awesome. In fact, many reptile taxa over evolutionary time switch back and forth. And so sometimes they will use chromosomes to determine their sex, and then over evolutionary time, their descendants would switch to this environmental determination of sex, and then their descendants switch back to chromosome determination of sex. Sweet. So here's that fish example that I talked about earlier. And this is an example of how some fish can use clues in their environment to determine what sex they're going to be. So this is Genecanthus semifasciatus. Okay? They're all born female. This is called protogyny. Proto meaning first, gyne being female, first females. So all the fish when they hatch are female. And then they live in these groups where you got lots of females and one male. If the male in their group is removed, then the biggest female turns into a male. So that's how they're using cues from their environment to determine their sex, right? It depends upon whether or not there's another male. And if you're the biggest female and your male goes, now that's a clue to you to switch to male. Pretty cool. This is actually a factor that we're concerned about with several fish species because, like I think red drum, anglers always want to catch the biggest fish, and so they tend to catch the males, and so they're constantly removing that male, and so the biggest female is constantly having to create a new male from a female, but that takes time. It actually doesn't take all that much time. A lot of times, within 24 hours, the biggest female changes color and starts courting the other females, and within a couple of weeks, that female has grown testes and has become a male. But still, if you keep doing this, you keep pulling those males out, you can have reduced overall reproduction because it does take a while. But don't detract that from the fact that how cool is this, right? That they can just switch whenever that male is taken away. A lot of gobies in the family Gobiidae can switch back and forth. And so based upon, again, the social environment and how many males and females there are, they can switch from male to female and then back again. So, again, really cool. Now here's an example, a picture, a friend of mine that I went to school with, he was studying sturgeon on the Mississippi River, and that's what this fish is. So you're looking at the ventral surface of a sturgeon that's been split open so that we can look at the gonads. And what you're seeing here is the white is testes, and the dark spots, those are eggs. These are sturgeon, right? This is where you get caviar from. So if you've ever seen caviar, you know the little black things are the eggs. And so you've got these hermaphroditic gonads. You've got eggs growing in testes, but they're not supposed to, right? There are lots of species that are simultaneous hermaphrodites. This ain't one of them. And so that's why he was studying them, because this is not supposed to happen. Probably this is due to chemicals in the water. There's lots of herbicides and other chemicals that we spray on land, and then it eventually washes down into the water. And a lot of these herbicides in that, they work by mimicking hormones. And the problem is then when they get into the water, they can still mimic hormones, and we think that that's what's causing this hermaphroditism in the sturgeon. And this then reduces their reproductive output because, their gonads are not quite right. And this is a species that's already in lots of trouble. Okay, let's briefly talk about the origin of gametes. We've talked about meiosis, but specifically, like, where does this occur and how do those gametes, how does the egg and sperm get formed? Excuse me. So spermatogenesis is the formation of sperm, oogenesis is the formation of eggs. And so this is in a human male. There's a figure from your book, kind of showing in the testes where the sperm are produced and how that relates to meiosis. And so if you kind of follow the sequence and kind of zooming in further and further here, and so you're looking at one testes here, and then inside that you've got the seminiferous tubules coiled up inside the testes. And so then now, if you look in this figure, we're zooming in and looking at a cross section of one of those tubules. So you got this long tube, and we're looking, kind of cut it and look at the end of that tube, and then zooming way in on that. And so at the outer edge, the outer part of that tubule, you have cells, and they're undergoing mitosis, right? They need to keep making new copies of those original cells. But then every once in a while, one of those cells will start to undergo meiosis. And they start to undergo meiosis as they migrate toward the center of the tube. And so they start out at the edge, and as they move toward the center of the tube, that kicks off meiosis. And as you get closer and closer to the center of the tube, you're further and further along in meiosis, to when you get to the center of the tube, then meiosis completes and you've got a sperm, and it's now in the center of that tube, so it can move down that tube and get collected and form ejaculate. And so it travels down the center of that seminiferous tubule because now it's a full-fledged sperm. And again, sperm are motile, they can move, they're very small, there's not much to them. They're mostly just DNA and mitochondria, right? Why mitochondria? Well, if you're motile, if you're, you got to make that flagellum move, you need energy, you need ATP, and so you need a way to make ATP, so they have lots of mitochondria, but then just DNA, and there's not much else in there. At the tip of this sperm, you see this known as the acrosome. The acrosome has enzymes that allow it to penetrate and eat through the outer layers of the egg. And so once that acrosome contacts an egg, then the enzymes can get to work and chew a hole, basically, through there, so then the DNA can be injected in. And what's kind of cool is that this process sort of changes the polarity of the membrane, and once one sperm has attached, then the membrane changes rapidly, and so then other sperms, the acrosome doesn't work anymore. So it's a way to ensure that only one copy of the male's DNA gets into the egg. Okay, and so this is a figure of the human female reproductive system. And you can see that the ovaries are attached here, and then that's where the eggs are produced, and then they're released and travel down the fallopian tubes to get to the uterus. Fallopian tubes are also called the oviducts, right? A duct for eggs. So here's another figure from your book, zooming in on those ovaries, which is where meiosis takes place and the egg is formed. Now what's interesting is that as that egg is formed, if you look over here on the left, it's showing oogenesis, and it's showing the steps of meiosis, right, that you're familiar with. But what they're trying to show you here, if you notice, that there's an unequal parceling of the cytoplasm. And so we're used to meiosis where you have that first cytokinesis, and you've got 2 cells, and then each of those goes on cytokinesis, and then you've got 4 cells. But what they're showing you here is with eggs, they're not equal. That during the end of meiosis 1, you have cytokinesis, but one of the cells gets most of the cytoplasm, and the other one hardly gets any. And that small one is called a polar body. It just has DNA. Now that one big cell that's left, then it continues through meiosis 2. Again, you have cytokinesis, and again it's unequal, to where only one of the remaining cells gets most of the cytoplasm, and you have another polar body. And these polar bodies never develop. They're just mostly DNA. They just get destroyed and reabsorbed. And so in this way, you can, instead of having 4 eggs from every bout of meiosis, you only get 1, but it's a lot bigger. It gets a lot more cytoplasm. And again, you remember the eggs are big and non-motile, but there's an advantage to that. And this is how you get those big eggs. They get all that cytoplasm. Now in humans, those eggs are all formed before you're born. So you're born with all the eggs already produced, but they're all paused in prophase 1. And they stay that way until puberty. And then once you hit puberty, then once a month, one of them will come out of prophase 1, complete meiosis 1, complete the first stage of meiosis, and so then you have that haploid egg. But it's still got double the DNA, right? Then, if that egg gets fertilized, when that sperm hits the egg, that stimulates the egg to go through meiosis 2, kick out 1 copy of the female's DNA, and now you've got that fertilized zygote that's got half the dad's DNA and half the mom's DNA. So again, that's interesting, that females are all born with all the eggs they're ever going to have, and then they just release 1 a month, and it's paused in meiosis 1 until it gets fertilized. Paused at the end of meiosis 1 until it gets fertilized. Well, this happens in lots of organisms. So for example, fish, again, I'm a fish guy, that egg is sitting there at the end of meiosis 2, and it still has 2 copies of the mother's DNA, and that sperm comes in and brings in a copy of the male's DNA. So for a split second, that zygote is triploid. Now typically, 1 copy of the mother's DNA, one of those polar bodies, gets ejected, and you're back down to diploid, and that's how you develop. And the same thing happens in humans. But if you shock the egg at just that moment, it won't eject that polar body. It will retain that polar body. And so now you've got a triploid organism. And so this is how we can produce like triploid fish. And remember about triploid fish, they got an odd number of chromosomes, so they're sterile, but they can still be viable. And so if you've got a fish that you want, to grow to eat grass or something, but you don't want it to reproduce, this is a good way to produce those. This is another also how you can get diploid offspring from parthenogenesis, right? We said that if you develop an unfertilized egg, at some point you have to double the DNA to get it back to the diploid state. Well, this is one way that you can do that. If you don't eject that polar body, then that egg is diploid and can develop parthenogenically. So again, it's fascinating all the different ways that animals can reproduce. Okay. Last thing I want to do is just some vocabulary. 1 slide. These are just terms that you need to be familiar with. Oviparous means egg birth. And so these are organisms, these are animals that lay eggs, right? Now the egg can be either fertilized inside the female or outside the female. And so you can have external fertilization like in fish. So the female lays the egg and then the male fertilizes it. Or you can have internal fertilization like with birds, right? The male fertilizes the egg inside the female, but then she lays the egg. That's oviparous. You can have ovoviparous, which is when the egg is not laid, but it's retained inside the female. So you're still producing an egg, and the embryo is still nourished by the yolk of that egg, but instead of laying it, like, laying it in a nest and then letting it develop outside the female, it's still developed inside the female. So it's still got some level of protection, but she's not nourishing it directly. The yolk of the egg is still nourishing it. And finally, you've got viviparous, or live birth. And that's when the egg develops in the uterus or an oviduct in some species. And so you retain the egg completely inside the female, and then the embryo is nourished directly by the mother. And so, for example, in humans, we're placental mammals, and so the embryo is nourished by a placenta. In marsupials, the embryo develops, and then it's born, and then it still finishes development in the mother's pouch, and so on. So anyway, these are just terms that you need to be familiar with, and that's all I'm going to say about that. So anyway, so that's our brief introduction into reproductive patterns in animals. Again, I think it's fascinating stuff. Just shows you that there's an incredible spectrum, and there's no typical way to form sex or to form offspring, and that's what makes biology so cool. So let me know if you got any questions, and I'll talk to you later. See ya.